first thing I want to say is, because I always forget to do this at the end, is to thank ASB&B and the student chapters for putting this together. All of you for listening and caring about our students. And of course, as Teo mentioned, a lot of the work that we're doing is funded by NSF. So, I always like to start any talk with this slide. And this year it seemed particularly appropriate. And in fact, in the middle of early March, when we switched from in-person to virtual, I showed the students this for a second time. So I'd shown them at the start of class. And we decided that we were all going to do the best of learning at the edge of chaos through the semester. So just a quick background to what I want to cover this afternoon. I'm going to give you a very brief background to cures. I suspect most of you are familiar with that. I'm going to talk a little bit about the length of a cure. You can do them in full semester, or you can do them as a module within a semester. But the main thing I want to talk about today is adapting a cure and adapting a course with a cure to using computational approaches. And that will focus on what questions do you want to ask, how to organize the lab periods, and a little bit of discussion about integrating with available wet lab approaches. If you're teaching in a hybrid format where you can actually do some wet lab, you know, the combination with virtual and computational approaches can be really powerful. I'm also at the very end of my segment of the talk, I'm going to talk a little bit about how to get involved with an ongoing project, though everything I'm going to talk about with the computational approaches can be applied to virtually any protein you choose. The last segment of the talk is going to be first Betsy Martinez-Vass and Tamara Manns are going to talk about how to build collaboration into a cure setting in a lab. And then Tony Bell is going to talk a little bit about adapting the computational approaches to any protein that you want to use. So the course I teach, the lab course I teach, had 5 specific learning goals. And these are probably fairly typical of lab courses. Talks about using the primary literature, some conceptual focus, which in my case was non-covalent interactions, skills such as keeping an accurate lab notebook. But in a cure, these 2 tend to be the sort of leading aspects of the course, and that is developing the ability to develop a hypothesis and produce some sort of research proposal, and then design and perform experiments to explore that hypothesis. And then the final learning goal of the course was to use your results to talk about your hypothesis and draw evidence-based conclusions. And then in the learning goals, it said obtain from a variety of biochemical and biophysical techniques to explore protein structure function relationships. So a cure really consists of 3 blocks of components. The first I like to think about is ending in a proposal, and you address issues of relevant scientific background and hypothesis development. The second block is where you do the experiments, and often involves teamwork, collaboration. I always try to emphasize reproducibility. I ask the students towards the latter part of the semester, pick the experiment that you've done so far that you think is the most important in respect to your hypothesis, and reproduce it, so that they're always thinking about reproducibility as well. And then, of course, as we talked about, data analysis and drawing evidence-based conclusions. The semester, or the cure, always ends up with some sort of presentation. It can be written, it can be oral, it can be a poster, it can be a combination of all three. The course I teach is a writing intensive course, so it actually involves all three components in that final presentation. But again, depending on the circumstances and what your course is trying to do, you can use some sort of presentation at the end of the course. So the second issue I want to address is, how long is a cure? Well, traditionally, people have thought of them as a full semester, and the whole square here would be the full semester. And then I've indicated on the y-axis what sort of things you would break down segments of the semester into. A modular cure is where you only use maybe a 5-6 week period of the semester. Maybe you've got other things you need to expose the students to, maybe requirements that the department has for them to see certain techniques or whatever. So a modular cure has all the same elements as a cure, but fits into a 5 or 6 week time frame. Now, under our IUS grant, we've been studying the impact of full semester versus modular cures. And it turns out, at least the preliminary data, shows that a modular cure carries many of the benefits that a full semester cure does. And that makes it much easier to adapt a cure into an existing course. So this is sort of an overview of how the course is set up and what changed and what didn't change when we made that switch due to COVID. So we always start off talking about big picture, what do you want to understand? That leads into background, preliminary observations, what is currently known, existing knowledge, and what is not known, what's the focus of where you want to take your experiments. Students then develop a hypothesis, and the hypothesis, I always have to remind them this, the hypothesis is really built around what you think is happening in the normal situation. That's followed by predictions. You know, if I change X, what will happen? And then we have a session on questions, a section on questions. What have you got to measure to look at these predictions? Then we talk about experimental approaches, the how can I measure it? And obviously this was the part that changed when we moved to COVID to online. Though as I'll try to convince you, the questions and the predictions and everything else on here really didn't have to change at all. And we'll talk about that as we go through this presentation. So just to give you a rough idea of what a full semester would look like in terms of number of weeks, this is translating that box into statements. We have a variety of student assessments that go throughout the semester. As Erica talked about, using lots of sort of low stake assessments, both formative and summative throughout the semester, really helps allay student fears. And then in a modular cure, you can put it wherever you like in the semester. But again, you break it down into those components that a cure has, life runs fairly smoothly. So COVID happened early March. We had to switch. The lab I teach was a Tuesday, Thursday lab. We taught in person on a Thursday, we taught in person on the Tuesday. The Thursday, we were online. And so we had basically 48 hours to think about how we were going to structure this. what we were going to do in a virtual world. We needed starting materials. We needed to prepare the starting materials and characterize the starting materials. We needed to talk about how the questions that we'd started in the wet lab phase translated into the virtual world. Then we did some preliminary experiments with some mutants to help refine our ideas. And all of this was done using free web-based servers around the world. And we collected lots and lots of data from that point on. And I'll go into a little bit of detail as we go through this presentation. The good thing is we were able to collect multiple sets of data. Students could do all the averaging and statistical analysis that we wanted them to do. In this case, we were comparing wild type and mutants, computationally produced wild type and mutants, and wherever possible, we put that into the context of what information might be available in the literature. The other thing we did was every lab period, we had mini group meetings where we talked about the aims, we talked about the data, we talked about the approaches. And one of the rules was everybody had to talk. I try to structure the class so that by the end of the semester, where I want them to be talking and presenting, the fear factor of talking in front of a group of students and often a group of other faculty as well had disappeared. And so we tried to build that in throughout the semester. Okay, so I'm going to talk a lot about computational techniques in a minute, but for each of them, we introduce them with a journal club about the computational approach and the type of information it could give, how it worked. And this was the big one. We discussed the validity and the limitations. And actually the students always had that question, well, how do we know this is a valid approach? And it was a really good way to engage the students in the, understanding the approach. We usually set up computations at the end of a lab period. And the reason for that is many of the computations you submit to a server and you get the results back in 24 hours. So it didn't lend itself to in-class computations. There are some of the techniques where we could do things in real time, and I'll point that out as I summarize the techniques. But then at the start of the next lab, we walked through the data analysis, and then each student spent time analyzing their data. Now you'll notice I highlighted we in every one of these. One of the things I found that really made this work and really kept the students fully engaged was this we. I was doing the experiments alongside them. When they were doing a computation, I was doing a computation. So in real time, this was all taught synchronously. In real time, we were able to interact with one another. If a problem cropped up, students, I was in the middle of using the same technique. I could help troubleshoot, or they could sometimes help me troubleshoot. But it became a group activity to do the experiments. And same with the presentations. We all talked about the data, what we thought it meant. Okay. At the end of each block of computations, we talked about lab experimental approaches that could be used to give complementary data. And then we also, because it was a writing course, they had to write up these approaches. We spent a little bit of time at the end of each block talking about how you would write up that particular computational approach, that particular type of data. Okay, so what did we do? Well, we used a variety of techniques where we could ask questions, and we could ask questions about conformation of proteins. We could ask questions about protein-protein interactions. We could ask questions about ligand binding. We could ask questions about potential and cryptic sites on proteins. And these are the approaches. The ones in purple are the ones where we could actually do the experiment in real time in the class. The ones in red are the ones where we had to prep at the end of a class to get the data back to look at the following class. This all started with doing some bioinformatics and using things like Clustal Omega. And the students loved making these sort of web logos of conservation, and it gave them something visual that they could talk about. Main thing was, all of these techniques gave us lots and lots of quantitative data. So what are the techniques? Well, the first thing we had to do was prepare proteins. There are two ways you can prepare a protein. If there's a structure in the protein database, you can just grab the PDB file from the protein database. If there isn't a structure in the protein database, you can construct a homology model. We use Fire2, there are a variety, but Fire2 is sort of a particularly easy one to use. So we use this, and you just submit the sequence. The cool thing is, if you want to make a mutant, you just change the amino acid in the sequence and submit the mutant sequence. And the students quickly got used to the fact that they could actually make 19 mutations of any given amino acid, and they could get the PDB files back from Fire before the next lab. So this was a real advantage. It let them explore different aspects of mutations by making multiple mutants of the same position. Okay, they could make the mutants in PyMol as well. You can go into PyMol and make a mutation. The trouble with doing it in PyMol is that it doesn't energy minimize the structure. Fire does that to a certain extent, but we got into the habit of using an energy minimization to refine the resultant structures, whether it came from Fire or from PyMol, and we use this program Refine-D. That does a great job, because it not only refines the structure, but it gives you multiple potential conformations. So you've got built-in 5 sample sets you can start collecting if you want to average data and things like that. The final thing we did was we used mole probity to get the statistics on the structure to characterize the structure. It gave you a Ramachandran plot. It gave you all the statistics on the structure. So what I should say now, so that I don't forget to say this later as I go through the other techniques, we've made tutorials on all of these, all of the techniques that I'm going to be talking about today, of how to use the servers that we've listed here. And those you can go in and download the tutorials. And these are basically hands-on how to do these things. So those processes make monomeric proteins. Well, sometimes you want oligomeric proteins, and you might want a homooligomer or a heteropolymer or whatever, and you can make those. So this is a dimeric protein made from subunits that we got from Fire and then assembled into a dimer where there was an existing crystal structure of homologous protein, and you could make it in PyMol. If you don't have a structure of the complex, you can actually construct a potential structure in this program called HawkDoc. And this is an assembly of a dimer with a homodimer with a different protein, in this case citrate synthase and malate dehydrogenase. You can build a structure of the potential metabolon. You can put in restraints. If you have some biochemical information about what things are close to one another, you can put those restraints in and get a model. Whether it's the correct model, of course, depends upon somebody crystallizing the complex, but that's a different problem. So once you've constructed the proteins, what can you do with them? So I'm going to do these in the order we use them in the course. This program, H++, is a really cool program. It's out of Virginia Tech. It was developed there, and the server resides at Virginia Tech. But what it does is it calculates the pKa values of protonatal side chains from a structure. What you submit the PDB file that you just finished making and characterizing. What you get back is you get pKa values for every protonatable group in the protein. And this is the cool part, it tells you which residues contribute to those pKs not being the pKs that you read about in the textbook. In other words, histidine is rarely 6.0 in a real structure, and this individual pK for histidine is not 6.0 most of the time, but you get a list of what contributes to it not being 6.0 for each individual histidine in the structure or whatever. So what can you do with this information? Well, a whole bunch of different types of experiments. You can explore the impact of an interface on an oligomeric structure on pKa values. You can relate changes in pKa values of mechanism-related groups, for instance, if there's a catalytic base in the protein, to potential changes in activity, either catalytic or binding or whatever. You can compare mutants and wild-type proteins. And you get data that you can put into nice plots. In this case, this is delta pKa of a mutation of a protein, comparing how much it changed with the non-mutated protein. So H++, great program. You can use it in real time. So if you have a protein that's oligomeric, or you want to make or look at oligomeric protein-protein interactions, you can quantitate those interactions using this program called HawkDoc, and in particular, one of the modules within HawkDoc. This, like H++, is something where you can get data back in about 10, 15 minutes from submitting it. Again, you submit the PDB file of the oligomer, you tell it which is the A subunit and B subunit or whatever. What you get back is the total energy of interaction between the designated subunits. You get the residue by residue contributions. And of course, it's always appropriate to point out, and you can see in this table, the red ones, this is typical output. So you have some unfavorable interactions and you have favorable interactions along the interface. This is the residues in the interface and the favorable or unfavorable interactions. And then if you make mutants, you can compare, these are the delta Gs for individual residues. So you can see which residues change, etc. So, you can explore the nature of the interaction, how mutations might affect the interface. You can compare isoforms, things like that. So this is a really cool program that we use during the course of the semester. It's got the name POCASA. I've never quite worked out what POCASA actually stands for. But again, it's a program that you can do in real time. What POCASA does is it searches for cavities on the surface of a protein. You can complement that by using SwissDoc, which docks designated ligands to a target protein. And I'll give you an example of that in a few moments. Again, what do you submit? You submit the PDB file for POCASA. For SwissDoc, you have to submit a PDB file of both the protein, either of these can be monomeric or oligomeric or whatever you like, and the ligand structure PDB file. You can often find that in what's called the Zinc database of ligands, small molecule ligands, or you can upload it. You can make it in a program and upload it. What you get back, POCASA gives you the location, area, and depth information of the cavities on the surface of the protein. You can use different probe sizes. So up here is a picture of the cavities colored by depth. So the shallow ones are the lightest color to the deepest ones, which are the darkest color. So this is the active site region. This is a cryptic site outside the active site. And so you can explore the structure of the protein. You can make mutations and see how it changes, et cetera, et cetera. With SwissDoc, what you get back is you get back thermodynamic information and location on binding sites on the surface of the protein. So if any of you are familiar with Carla Matos at Boston University, a number of years ago, she developed a technique called multiple solvent crystal structures. And it turns out this SwissDoc application is a computational version of that. You can give it different ligands and it will map out the sites on the protein where those ligands bind. You can look at the thermodynamics and tell where the high affinity and low affinity sites are. And if you select ligands so that they're sort of fragments of biological ligands, you can sort of map out potential sites from their sort of chemical specificity rather than just their structural. So again, you can use these to look at a whole bunch of different questions. So the next technique that I want to talk about is using SwissDoc in a slightly different way. And this is now, what SwissDoc does. As we talked about, it gives you docking of a ligand to a protein. Blind docking is where you don't tell it where on the protein you want to find the sites, and it finds them all over the place. Local docking is where you have an idea where the active site is. So you literally draw a box around the active site and say, I only care about the binding modes that are in the active site. If you want to design a mutation that you think might affect affinity of a ligand, this is a great way to examine that. Again, you give it the PDB file, etc. What you get back is structures of these poses of the ligand in the active site, and there are many poses here shown. But you get the thermodynamics of all of the poses, okay? And it breaks it down into all the components that are involved in ligand binding. So it's a great way to talk about what actually is involved in ligand binding to a protein. So I mentioned that we did a journal club. These are the papers we used. There's a paper here for each technique. There's the links to PubMed. They're all, I think, free off PubMed. If you have any trouble finding them, let me know and I'll make them available to you, but they're all there. So the final thing I want to say is that I teach based upon this thing called malate dehydrogenase, an enzyme everybody on this Zoom has heard about somewhere. This is the malate dehydrogenase cures community, which is the title of our NSF-funded program. Tony is going to talk about how you can adapt the techniques to other proteins easily. But if you want to join, if you want a sort of off-the-shelf project on malate dehydrogenase, let me know. Email me. We've got 2 ongoing projects that are multi-institution projects. One involves the active site region, one involves the subunit interfaces. There are other projects always coming online. So let me know if you'd like to join those. The other thing, in 45 minutes, which is the time allotted here, we really can't go into the sort of hands-on use of all of these techniques. As part of our NSF grant on Thursday, August the 6th and Friday, August the 7th, we're going to run hands-on training sessions where you'll actually step through these, all these computational techniques, or any of them that you think you might want to have hands-on experience of before you put them into your class. And if you want to join in with that, email here and we'll send you the Zoom link so you can join in these sessions. So I think I had promised Betsy I would finish somewhere, Tam, I would finish somewhere between 10:30 and 10:33. I think I've made it. So it's my pleasure and privilege to introduce Tamara Mans and Betsy Martinez-Bass, who are two members of the MCC and are great colleagues to work with. So I'm going to quit sharing my screen so that you guys can take over.
Okay. Hello, I'm Tam Mans. I'm not 100% sure if Betsy got in. I know she was having a little trouble. I'll get started. I know she had some lovely, oh, I see her. And she has slides. Is it possible to show the slides, Betsy?
Yes, I'm trying to share the slides right now.
Okay, great. While she's working on that, I'll just introduce myself. So I teach at North Hennepin Community College, and that's in the northwest corner of the Twin Cities. We're an open enrollment institution. 62% of our students are considered underrepresented. And I'm using a cure that concerns enzyme structure and function, and I use it in my biology 1101 class. It's called Principles of Bio 1. I use it in the lab. That's the first biology course for our science majors. The only prereq for the class is that they've either taken or are currently enrolled in Chem 1. So they're very introductory students.
Oh, hi. My name is Betsy Martinez-Bass, and I work at Hamline University in St. Paul, Minnesota. So Hamline is a mid-sized university, but in terms of the sciences, we are considered a primarily undergraduate institution. The number of students that we have that can be classified as underrepresented minorities or first-generation students is up to 40% of our student population at the time. And when I was collaborating with Tam in this cure, I was working with the students who are in the Biochemistry 1 class at Hamline. So I am going to tell you, I've been trying to share my slides, but I cannot get Zoom to work from my computer. And then, on my phone, I've been trying, but, it's really not working. So my apologies in advance, but I think we can tell you about what we did. And first, I want to start by telling you about how we set up our collaboration. Where basically Tam was teaching her introductory biology class at North Hennepin Community College, and I was teaching biochemistry at Hamline. So the very first step for our collaboration was to meet, and we selected loop mutants because Tam had worked on the loop mutant project with Ellis and his group. And Tam was very effective and very helpful, helping me plan assignments and use some of the assignments that she had used in the past. So that was the first part. We met, we selected some mutants for the students to work. And after that, we work on getting the students, both of our groups to work on hypothesis testing and experimental planning. And that was really nice. And then we scheduled classroom visits. So we had classroom visits early during the semester, and I visited Tam's class and listened to her students present about their hypotheses and the mutants they have selected. And then Tam came to my class and did the same thing. So then following that, what Hamline students did, because the Hamline students were taking biochemistry and they learned how to purify protein, my students purify a lot of protein, wild-type MDH for Tam. And then I delivered that wild-type MDH for Tam to North Hennepin. And I also visited her class a couple times, as I said before. So Tam students got to test the protein that my students purify. And we have mixed success. We have, protein that was fantastic, very active. And then we had some other proteins that were not very good and wouldn't work. So that was a good opportunity for our students, to talk about, oh, your mutant was good, or your wild type was good or not good, etc. Tam, would you like to add something else to the setup?
Yeah, I think you did a good job. Basically, yeah. So we went back and forth a little bit. So her, well, I got to go to her classroom and listen to her students talk about their hypothesis. I could ask them questions about what they were thinking, sort of make them explain a little more about their hypothesis. And then she did a similar thing with my students. And in case you're wondering, would this work virtually? Yes, we've done this virtually in other collaborations where, you know, I haven't been able to actually go to someone's room, but I can, you know, do a Zoom call or something like that. And the students take their presentations a lot more seriously if it's involving just one person from outside of their classroom. So even just having one faculty member that's not their primary instructor, it made a huge difference. They took it a lot more seriously. And also, I thought it was fun that Betsy's students thought I was some, you know, expert on this topic. And I didn't feel like an expert until at the end of the class when they were like, oh my goodness, we want to know your opinion about this. Why were you asking that particular question? So that was fun to visit each other's rooms and kind of see how it went. And then our students were also communicating mostly virtually, actually, even though they could have like picked up the phone, they preferred to do chats and stuff like that.
So I wanted to add that my students and TAM students had an assignment regarding a reflection about the QR and what they learned about the QR. And I was able to read through it, and I had created a word cloud that I was going to show you. Tam, I just forwarded you the slides. So if by any chance you got them, maybe you can put the word cloud up. But if you didn't, I can tell you that the main words that we can see in all the students' comments are words like collaboration, difference, support, scientific research, discussing things, finding new solutions for problems, etc. So I have a set of quotes, and I was planning on showing them to you, but I had some technical difficulties. But I can read some of them to you. So one of my favorite ones is the following quote from one of my students. This collaboration was a great way to experience how research might be done in the real world. It showed how collaboration between different institutions might be beneficial in finding the answer that might be of interest to both parties. So this is a good aspect. They learn how collaborations work in the real world. In addition to that, here is another of my favorite quotes that says, it can be hard to come to terms with failing, but also hard to realize that results aren't right or wrong. We will run an experiment and present saying that it didn't work, but in fact, it did work. The results just weren't what we expected. It was interesting to learn that others share the same sentiment. And here's another quote that I really enjoy. We found that there are many different problems that can be faced when working on an experiment. All the groups have different experiences and problems and ways of solving these problems through trial and error. So, you know, my students and TAM students really enjoy working together and getting into contact, you know, as Tam said, mostly virtually over the phone and, you know, over email. I think next time when we do this, we probably invite students to visit our classes. But overall, it was great. And one of the things my students kept saying all the time was like, oh, wow, we are collaborating with a class of first-year students. It's wonderful that they get to work in such a complex project. And they were always very aware of the fact that they were communicating with students who were junior students with, you know, a little bit less experience. So they needed to be very explicit and make sure they were communicating effectively.
And I was going to say that I felt like my students felt like the work was truly important because someone outside of the classroom cared about what they did. And they also got that sense of how scientists really work together and that being part of that they were included in that community.
So I'm truly looking forward to working with Tam again in the near future. And this was a fabulous experience. So thank you.
And I think we're going to introduce Tony Bell, who is at the University of San Diego, and he's going to talk about how to use more than just malate dehydrogenase.
Right.
But in addition.
Yes. Thank you, Tam. Wow. Do you, let me see. Let me get out of this because I think you see my, jump in the PowerPoint. I thought I would work on Keynote, it didn't.
Maybe sometimes it helps if you just do the share screen instead of share PowerPoint.
Yeah, I'm looking for my, okay, here we go. Okay, so do y'all see the screen or my notes?
Notes.
Notes? All right.
But we can
Okay.
we can get it, Tone. Go for it.
Look at my notes then. All right. So I was actually teaching the Monday, Wednesday section in the Biochem lab at USD. So when Ellis said things that meant we, he probably should have put we and them, because he was often serving as IT as I was texting him when we were having difficulty with our stuff. But it all worked out. And the one thing about this is that I'm the third bell in the USD biochemistry department, and I did not have any knowledge of MDH before I showed up here. So we got to working on this, and Ellis was always asking, like, what do you want to do, you know, with this type of project on something that's in line with your research. So the protein that I work with is a protein called HMGB1. So it's superior to MDH because it has one more letter and a number. So HMGB1 stands for high mobility group B protein. And in terms of the research and learning objectives, it's that we want to use all of the things that we did in the biochem lab to help discover new cellular binding partners for HMGB1. So it's a lot of initials, but what this protein is that it's a multifunctional protein that's abundant. It's about 1 million copies per cell. It's found in the nucleus. And when it's in the nucleus, it's a DNA chaperone. So it essentially binds to bent DNA in a non-sequence specific manner, and it promotes transcription factor binding, DNA repair, and recombination. In grad school, I was more interested in this kind of realm of the protein, but as time has gone on, I'm more interested in what's been discovered as the extracellular role of this protein, is that it can be actively released by immune cells or passively released by all cells. And when it enters the extracellular matrix, it binds to immune receptors and it can trigger pro-inflammatory cytokine reactions. What I'm more interested in is an understudied function of this extracellular role of the protein. And that's that hypotheses have shown is that, or experiments have shown, is that it binds to cell clearance molecules, particularly phosphatidylserine, which is a cell surface receptor, which apoptotic cells use to signal for macrophages to come in and eat them. And it binds to the receptor on macrophages that link these together. And it also binds to adapter molecules that put dying cells and macrophages in close proximity to each other. And our hypothesis is that this highly abundant protein, that's a multifunctional binding protein, more than likely binds to many more cell clearance molecules beyond the ones that have been already characterized. And so that's what brings us to merging my research endeavors with what we did in the lab. And that is to use computational techniques to help discover new cell clearance binding partners for this protein. And we're doing some of the same things, or hope to do some of the same things that we set up in the lab. And that is using SwissDoc, using UCF Chimera, and PyMol to essentially dock known small ligands to the protein, and then use Chimera to look at the binding energy of certain ligands to then attempt to go back in the PyMol and measure the binding distances to that. Because the one caveat about UCF Chimera is that you have to use the free files that you get from SwissDoc within about a week, or they magically go away. We found this out the hard way in class, but find all your poses and do everything you want in Chimera, and then you can go back later and work in PyMol as long as you want. So what I didn't mention in the background is that this protein has 3 subunits, and 2 of them have PDB structures that have been solved. So the idea is to use SwissDoc to determine the computational conformers for known ligands that bind to this thing, and just basically try to identify a universal binding motif. And essentially, these are the controls, because we know that small molecules bind to certain subunits of this protein. And another good thing about this is that although 2 subunits are already known, the full-length protein has not been solved. So we can use FIRE2 to generate a PDB model of that. And then we can go back through the SwissDoc and UCF Chimera and the PyMol iterations again and compare that data with what we've seen before to see if the full length protein has any different type of binding conformers to the controls, or what we're calling the controls there. And then from that, we would like to jump off into measuring protein-protein binding interactions after we've thoroughly exhausted the small molecule portions of that. And again, we can use the solved subunit structures for that to get the controls moving. And then after that, we can again use FIRE2 to build a model of the complete protein to then compare back to this. And we've already started doing some of this stuff in the lab. And this basically just shows a chimera pose of one of the subunits of the protein binding to a known small molecule that's been published. So the one thing that I feared when we switched into the virtual world and doing this stuff now is that, would you have enough, experiments to fill a semester or whatever, block of time? And at least with us, the answer is yes, because with SwissDoc, you can go through, essentially a library of small molecules. And also, one thing that Ellis had mentioned earlier is that you can also attempt to identify unknown binding motifs. So this protein is multifunctional, it binds to DNA. There's a ton of DNA protein docking software out there. So we could jump off in that direction too, to compare if you can find motifs that bind small molecules, proteins, and nucleic acids. So at first it seemed a little daunting, but I mean, essentially with FIRE2 and building models from unknown structures, I think it's not limitless, but I certainly think there's plenty to keep you busy, and it's certainly, I think, going to keep us busy for a while. But that was kind of my spiel in taking these virtual tools and adapting them to a protein that's not malate dehydrogenase, even though I've come to like malate dehydrogenase.